Okay, now to continue the conversation of weeds, we have Dr. Gage. She is an associate professor of weed science with a cross appointment in the School of Agriculture and Biological Sciences at Southern Illinois University, Carbondale. She earned her Ph.D. in plant biology with a focus on weed ecology from SIUC in 2013. Her research interests are centered around integrated weed management practices, which combine multiple tactics to build diversified weed management systems and control herbicide-resistant weeds. Today, we will hear from Dr. Gage as she presents on herbicide resistance, which reduces already limited herbicide options. The integration of other types of weed control tactics with chemical control programs have the potential to slow the development of herbicide resistance and steward available crop biotechnologies. This presentation will discuss integrated weed management and the benefits of adopting a diverse approach to weed control. Please help me welcome Dr. Gage to the 2022 Soybean Summit.
Thank you. Yeah, thanks so much for that welcome. It's such an honor to be here today to talk to you about my research and some of my thoughts around stewardship. So if I say I accidentally during the presentation, I mean the collective I. So all of these people fuel the program. Eric Miller is our weed science field program manager. He handles all the logistics for our field trials. Alexander Muth is a new researcher in my lab, and he's also going to be a grad student at the same time. Graduate students, Madison Decker, Mackenzie Trader, Bradley Decker, Samuel Ramirez, and Avery Sheik and I have all helped with the project that I'll present today, as well as numerous undergraduate student researchers that are in our lab every day. Ronald Kraus, the former farm manager at the Belleville Research Center, who many of you may know, has collaborated on some of the work that I'll tell you about today. And then I have reached out to multiple collaborators to get data from them. So today I'm going to show you some work that Dr. Kevin Bradley has done at the University of Missouri in collaboration with me and other weed scientists, Dr. Lauren Lazaro at Louisiana State University, and Dr. Prashant Jha at Iowa State University has kindly lent me some slides that are related to some of the things I'm going to show you today. And then, of course, the United Soybean Board. Some of the studies I'll show you today are funded by the United Soybean Board. I feel very lucky to be funded by the Soybean Board for the next fiscal year as well. I'm one of many scientists in two different projects that have been funded. One is led by Dr. Brian Young at Purdue University, and the other by Dr. Stephen Mirsky with USDA ARS. So I have to say that the results that I'm presenting today are my interpretation, and in some cases it's from other people's data, and it doesn't necessarily represent their thoughts or the thoughts of our funders. So this is what I want to tell you today. I want to tell you about the challenges that we're facing, and if you saw Dr. Aaron Hager's presentation, he covered those very well, and I'll reiterate some of the things that he talked about. But we know that stewardship is really important. We have to preserve the tools that we have. So I'll tell you about that. Then we'll talk about integrated weed management and why it matters. And then I'm going to cover a variety of integrated weed management practices. Our lab is involved in many more than just the studies I'll tell you about today, but I've picked just a couple, and they range from what is maybe comfortable and practical to things that are maybe a little less comfortable and more experimental. So I'll tell you about those things. And those are cover crops, interseeded crops, and then one particular harvest weed seed control tactic that is chaff lining. And then I'll end with just a couple of conclusions. So let's talk about challenges, and this we're going to start with an herbicide resistance update. So this is where Aaron gave a great overview of what we're facing in Illinois. But you can see the resistances that we have that are listed here on the slide, and I won't go through these. But in some fields with multiple resistant populations, it's getting really hard to find chemical management tactics that are working. So that's where we have to bring in novel ideas or innovative strategies to try to overcome these resistances that we're facing. And in this last one, so dicamba resistance was recently confirmed by Dr. Hager and his colleagues this past year in 2021. So this is Dr. Kevin Bradley's data at the University of Missouri. One of those United Soybean Board projects that I was telling you about is a collaborative study where multiple universities collect seed at harvest and send them in to different universities for screening. Dr. Bradley is handling the water hemp screening for all the states that you see with shaded counties here. So he receives all of our populations. He takes seed from each population and puts it in a flat that's about a foot by 2 feet, rectangular flat, He grows out those plants and then he sprays them with either a half-X rate of herbicide or a full rate. And you see the blue counties, there were no survivors in any of those replicates, any of those trays that he screened. Yellow counties, there were survivors in over one replicate. The red counties are where we're really concerned. That's where we had survivors in each replicate that he sprayed. So this is the kind of thing that keeps weed scientists up at night when we start talking about losing glufosinate. If you want to see those counties in Illinois, that kind of gives you an idea, zoomed in now, of what we're dealing with. The percent survival, the graphs that you see here, so this is a half-X rate of glufosinate, and this is a full rate. These are all of those counties that you saw averaged over all of those time periods. So the graph is not just Illinois data. I need to go in and look at the Illinois data. He sent that to both me and Aaron, so we can look at that. But we're looking at the average survival rate. So we see a half-X rate, you would expect to see survivors with a half-X. When we look at a full rate, though, this is the concerning part of the story here. So we have an increase in survivors to this full rate of glufosinate now in the greenhouse screen, and we can see this progressively ticking up over years. So this is something that we should all be watching out for. And then the same was done for dicamba. It was just these two herbicides for the previous years. The following year, this next year of seed, will be screened with 2,4-D as well. But you can see again, blue counties, no survivors. Red counties is where we potentially have a problem. And this is kind of fascinating to me. Our Illinois data does not look like this for a 1X rate. I'm looking forward to working with those data myself, but if you average across all counties, we actually see a progressive decline in survivors to dicamba. So I'll let you know next time we speak what that looks like for us in Southern Illinois. So stewardship and its importance. Well, the past couple of slides should basically speak to this by themselves, but we want to preserve all of the available tools that we have. Everything is so precious right now because we have so few tools available. We really want to take care of the resources that we have so that we can have them for as long as possible. So that's where stewardship comes in. And we know that one of the basic tenets of a good weed control program is a crop rotation. So what we do in our corn crop is helping with our weed control in soybeans. So we can't forget that rotational aspect is so important. One of the important things about that is our ability to rotate herbicides, right? We use different actives in corn than we do in soybeans. So remember that comes up in a minute. But we actually need more crops in rotation than just this. What we're selecting for by having a simplified rotation with only 2 crops, we're actually selecting for weeds that survive well in soybeans and weeds that survive well in corn. We're selecting for super competitors with a simplified 2-crop rotation. When we put 3 crops, and especially 4 crops in rotation, when we can find those other crops that make money and seem economically feasible, we see there are problems. We don't have the same problems with weed control and even pathogens in the field. So unfortunately, we don't have a lot of options economically. These are our most profitable crop options, and that's kind of where our rotational strategy goes. So at least we can rotate these 2 crops and do things differently in each crop. If we look at the soybean herbicides with Amaranthus activity, we have, and this goes back to our group numbers. So group number 14 are PPO inhibitors. You can see the actives there post-emergence. Photosystem 2 inhibitors, metribuzin and linuron, group 5 and 7. PPO inhibitors, again, group 14, pre-emerge. And group 15, long-chain fatty acid inhibitors. Those are very important. But again, you just heard from Aaron about now group 15 resistance that we're dealing with, which comes up in those overlapping residuals that we all know we need to be putting down. So when we factor in genetic modification, we can add a lot more site of action groups now to our management strategy. We can add in growth regulators. We can, of course, glyphosate, and you could say that's not effective anymore, and we could just take that off the list here. Glutamine synthesis inhibitors, so that's Liberty, right? And then PPO inhibitors again, and HPPD inhibitors. So now we're gaining some of the corn herbicides, right? The actives that we could use in corn, we can now use in soybean with the HPPD inhibitor trait. So how we steward that going forward is going to be critical, because now we're losing one of the valuable components of rotating those crops. So keep that in mind as well as a stewardship practice, and we need to steward those HPPD inhibitors really well. If we look at our pre-emerge actives, we've added here HPPD inhibitors again as a potential pre-emerge option. So what we want to do, we want to preserve these things. These genetically modified traits are the reason that we have a no tolerance threshold for weed control in genetically modified crops. If you see a weed escape in one of these crops, you should assume that you're dealing with a resistant weed. It's going to develop into a resistant population, and you're going to potentially lose that chemical tactic, that strategy. So we need to put other types of diversified management practices in with these chemical control strategies. That makes things a little bit messy. As an ecologist, I'm very familiar with all of these ecological interactions, and sometimes when you combine these things, you get unanticipated consequences, right? You don't get what you think you're going to get because there's some ecological interaction that happens that you didn't anticipate. So things get messy when you try to diversify your cropping system, but it's going to be critical moving forward to preserve the chemical tactics that we have. So that's where integrated weed management comes in. We have to switch things up so that we don't let the weeds adapt. We have to do things differently. And everything that we do, you can think of every management tactic that you put down in the field as something that is causing a shift in the genetics of that weed population. Even the cover crops that we plant, you can think of the weeds as adapting to the cover crops as well, which they will. And we could talk about that more if we have time. So what is integrated weed management? We could talk about it as a way to protect environmental, human, and economic well-being. We could also just simply consider it, for us, for our purposes, adding another management tactic with chemical control. And it could be any number of tactics. And so the GROW website, growiwm.org, Getting Rid of Weeds Through Integrated Weed Management, has some great resources on integrated weed management. We can start up here with chemical control, so we know herbicides and our crop trait systems are what we're talking about when we talk about chemical control. Mechanical, we can put harvest weed seed control practices in that category. Cultural control, we could talk about diversified rotations, narrow crop rows, planting densities, cover crops, and interseeded crops. And I'll talk to you about cover crops and interseeded crops today. Biological control, we don't have a lot of options in the Midwest. There are things that eat weed seeds, a good proportion of the weed seeds in the field that you may not even realize what kind of service they're doing. But if we look at things that we can actually introduce or market as biological control agents, we don't really have those in our soybean and corn cropping systems. And all of this sits on this, this prevention platform right here. So we have to do things like, you know, go manage our weed-free fields first. If you have a low-density field with not a lot of weed problems, you do everything in that field first so that you're not spreading seeds as you move through the landscape. So we have to think about things like that as well to prevent the movement of herbicide resistance across the landscape. So all of these things together, all of these diversified tactics make up this integrated weed management framework or theoretical concept. All of these things together, you could think of them as Ronnie Krause would say, as thinning the herd. So you have fewer individuals when it comes time to make that chemical application. And Dr. Hager told us it's a probabilities game, right? The more individuals you're applying that herbicide onto, the more likely you are to find one individual that has whatever genetic trait that's going to allow it to overcome that herbicide application. So we're trying to reduce the number of individuals that are being exposed to herbicide. So now we're going to talk about some things that are practical and some things that are a little bit more experimental that we're kind of working out. First thing I want to talk to you about is a cover crop system where we're rolling the cover crop and we're using a banded versus a blanket pre-emergent application. And this is in a cereal rye cover crop. So on the left, the picture that you see, we have a Dawn ZRX roller crimper planter. And it creates this really neat pattern here where we can open up the row and plant soybeans. It has a chevron-shaped pattern to the wheels, so it kind of pushes all the cover crop residue into the center between those rows, which creates a really thick, nice mat of biomass. So we're going to compare that with the system that you see on the right, the no cover crop system. And you can see the mare's tail already. You can see the dying mare's tail that was up in that system. So some of the specifications for this was, we've done this multiple years, but I'm going to show you 2020 data from Carbondale. We drilled cereal rye and balanza clover at 90 and 6 pounds, respectively. We planted enlist soybeans on 30-inch rows. We removed the cover crop biomass from about a 20-centimeter area over each row, and that allowed a 30% thicker mat of biomass between the rows. We used here, our pre was Valor XLT, and we applied that either as a blanket or a banded application over the top of the row. And then some plots, all plots received a post, a blanket post that was glyphosate plus 2,4-D. Some of those plots received a residual with that blanket post. And then we rated the field for weed control, and our targets were, of course, water hemp and mare's tail in this study. So I have to show you bar charts, sorry. I will explain what's happening here. So we have control on the y-axis, and this is water hemp here in red. This is mare's tail here in gray. And we have our treatments across the x-axis there. So first of all, no cover crop and non-treated. Of course, you wouldn't expect any control there. Cover crop non-treated, that's just what the cover crop is doing by itself. So 55% control with just a cover crop and no chemicals on top. No cover crop broadcast, so that's a fairly decent, that might be acceptable. Cover crop with a broadcast application is even better, of course, as you would expect. Cover crop banded here, we see a little dip in control, but all of these other treatments, whether we had a cover crop or not, broadcast post or banded post there, those all look good. And here, no cover crop or cover crop, broadcast post plus residual, all of those look good. So what you want to take away from this is that I didn't show you yield data, but I can tell you there was no difference in yield data. So our weed control and our yield data was comparable between those treatments. And that tells us that we could have a 65% reduction in herbicide usage with banded treatments in this system. So that's reducing the numbers of, reducing the acreage within that field that's receiving an herbicide application, and therefore the selection pressure. So now I'm going to talk to you about something that's a little bit more experimental. This is being worked out. We're still working on this system. Some years it looks great. Some years our interseeded crop does what it's supposed to do. It controls the weeds, it suppresses them, and the yield is no different than a soybean-only pre plus post system. But some years it hasn't looked quite as good. So I'm going to show you the worst case scenario, just so you see kind of what we're dealing with. But this is the interference of interseeded winter wheat on soybean growth and weed suppression. This is research that comes directly from some work that Dr. George Kapusta did at the Belleville Research Center a long time ago. So they had, he worked with Paul Myers. Paul Myers got his master's degree in 1983, and they studied the ability of giant foxtail to suppress broadleaf weeds in a soybean system. So they actually planted their soybeans into giant foxtail. If I stood here today and suggested that you do that, you would think I was crazy. So instead, we're working with winter wheat, which is so much easier to manage. So he saw that in 2 out of 3 studies, soybean yield was negatively impacted. And he was kind of working out the termination timing and kind of how that affected yield. But in the 3rd study, he found that there was no yield loss, and they attributed it to soil moisture availability. And that's what we see as well. So that's what I'll show you. So this was work that's done by a Ph.D. student, Madison Decker, who's finishing up her program. You can see here 0 days after treatment. So this is when we terminated the winter wheat, and you can see after termination there what it looks like. So first of all, we wanted to look at the optimal timing to plant winter wheat so that it could create enough biomass to suppress broadleaf weeds. So we looked at that, and then later I'll tell you in just a minute about the optimal winter wheat termination timing to try to preserve yield. So I'll show you data from it's conducted in 2019 and 2020. I'll show you the Belleville data. Winter wheat was planted at 1.5 million seeds per acre on 3 different dates, either prior to soybean planting in April, in early May or at the time of soybean planting in mid to late May. And then we had these herbicide treatments. So we had pre-applications of fumicipin plus S-metolachlor. We had, that was prefix followed by a post of dicamba and glyphosate, or we had post-only applications of dicamba plus glyphosate. We had an herbicide-free non-treated and a handweed non-treated as well. So this is just an outline of our planting dates: fall, April, early May, mid to late May, and then no wheat planted. So we had a soybean-only system as well. So we had non-treated, weed-free, post-only, and pre followed by post treatments here. So this is the mare's tail control from that planting timing study. So we didn't have a lot of biomass that developed in late May. This looks just, so this didn't establish. This looks just like our soybean-only treatment, really. Early May, there wasn't a lot of time for biomass development either. This April planting date looked really good here. And if you look at here, the purple is a non-treated. So that's what the winter wheat biomass alone is doing for water hemp control. And if you compare that to our pre followed by post in a soybean-only system, those are not statistically different from one another in their ability to control water hemp. So we could see water hemp suppression. But here's the yield data. So the post was applied at the same time. The planting date was different. Uniform post-application timing. So we see in our soybean-only system, we see relatively higher yields than in that April timing. So we know we need to work on our termination timing to see if we can do a better job preserving yield. So that brings us to the termination timing study now. So here are our treatments. Again, I'm going to show you, I'm going to talk about 2019 and 2020, same seeding rate. So we had a weedy and a weed-free non-treated and a post dicamba plus glyphosate, and we used 5 different soybean growth stages. The soybean-only plots had the same treatments. For comparison, we just wanted to do everything at the same timing to make sure that the timing of application wasn't a factor when we compared yield. So we had pre of sulfentrazone plus Grand Slam or Authority First. We had and dual magnum along with that, followed by dicamba and glyphosate at the V5 soybean growth stage. So here's our treatment list. Our timings here: V2, V3, V4, V5, V6, and then we had the weed-free non-treated, and we had our standard soybean comparison. So this is where it gets a little interesting. So the darker lines here are the 2020 rainfall events. The plants were not moisture stressed in 2020. Everything looked great. There were no differences in yield. All of those termination timings yielded really well. Everything looked great. But when we get to 2019, you can see it's like all the rain went around Belleville that season. It seems like so Madison even had to irrigate her research plots that year to just get the wheat to establish. So we had limited rainfall, and we can consider this kind of a worst case scenario. So these are the yield comparisons here. So you can see there's about a 30% yield hit comparing the soybean-only treatments. You could look at this pre followed by post. That's our real comparison here compared to the interseeding yields. So all this is saying is that, yes, in a year where you're moisture stressed, where the plants are stressed, that wheat is going to negatively impact yield. So I think there are ways that we can improve this system even still, and we're still working on that. But there's always a silver lining, right? We saw a difference in the soybean protein content here. And we know that competition can do that, but if you look at the protein content in the soybean-only pre followed by post-treatment here, it is significantly lower than all of our interseeded treatments here. So maybe better quality beans, even though the yield was less. So now, harvest weed seed control, we'll talk specifically about chaff lining and how it might be applicable in soybeans. So there are a number of different harvest weed seed control tactics. In another presentation, if I could do kind of like a sequel to this one, I'd tell you all about a trip that I took to Australia where I learned about 6 different types of harvest weed seed control that Australian growers are using. And that's because they also have such a horrible problem with herbicide resistance. So they're developing some pretty innovative non-chemical tactics. They have innovative growers that are coming forward and designing their own weed seed destruct systems. And you've heard of the Harrington seed destructor. We met Ray Harrington. He was an innovator that came up with this system and just kind of built it on his own. So chaff lining in particular is the lowest level of this harvest weed seed control, all of these tactics, because it's the cheapest. It's the most easily accessible. You don't need complicated machinery. It's not expensive to adapt combines to do this. And this has been widely adopted by growers in Australia. There's actually the first scientific publication that's out now about chaff lining in Australia. So this is an herbicide resistance management tool. It's easy to install. You see the chaff chute here on the back of the combine. There are no major modifications to the combine. You may have to add a baffle into the interior of the combine, but this is just, I'm told, a couple of bolts to add in this way that you separate the straw fraction from the chaff fraction. So those finest particulate matter of the soybean, that's harvested along with the seeds, and that's what goes into this chaff chute and out the back of the combine, and then the straw is broadcast on the field after that. So you're not losing organic matter, and that's one of the benefits of this system. So it kind of looks something like this. So this is in a small grain system here, and you see this most basic chaff chute here. It's just a way to funnel that chaff fraction into a line. This, if you know whose field this is, I would love to know who has this in Quincy, Illinois. Somebody's chaff lining in Quincy. You can see those lines here in the field. I thought that was really neat. So people are always watching. So this is what it looks like when annual ryegrass is emerging in chaff lines. This is actually a tram line. This is not really a true chaff line. So this is a way where you funnel the chaff behind the tires of the combine. And then every time you go into the field, you're basically running over your chaff lines. So that's chaff tram lining. But that's what you're seeing here. So you can clearly see the annual ryegrass is emerging in the lines. And already, I don't know how many years this has been going on, but you're not seeing the same level of density in the rest of the space in the field. And then, and this is a grower from Australia that's actually doing these practices here, Gary Lang. You see a banded application of herbicide on top of this chaff line. So think about this. Theoretically, you could bring in a hooded sprayer and apply something that's non-selective over the top of the chaff line. In Australian systems where livestock is incorporated, they actually take the livestock in and they graze the chaff lines before the weeds start producing seeds. So there's the ability to integrate some practices as well, even further there. So we're doing some pretty basic studies on chaff lining systems to try to figure out what that might look like in soybeans. This is a study that was led by Louisiana State University. I was part of this, as well as researchers from the University of Kentucky and University of Wisconsin-Madison. We also have a collaborator from Kansas State University on this as well. So we were simulating chaff lining in this study, and there were a couple of advantages to that. We could all standardize the amount of chaff that we were using. It wasn't dependent upon the header width or the combine type that we had. We could all standardize the amount of weed seeds that we put in those chaff lines as well. So we did this as a simulation to see what actually happened in that chaff line. We looked at weed seed viability over time, focusing on the Amaranthus species, which water hemp and Palmer amaranthus, what we were concerned with. So we actually combined this. We wanted to look at the interaction of some pre-emergent herbicides to see if there were any effects of that residue. So we were looking at the interaction here. We had, within the same plot, we had a line of weed seeds with chaff on top of it, which simulates what would actually happen as the seeds settle to the bottom because the chaff is lighter. And then we crossed those, we had the weed seeds and the chaff on top, and then we had a line that was just weed seeds alone, with no chaff on top, so that we could see the differences in emergence and how the chaff line might suppress that or enhance it. So, our treatments here, we had No-Pre, we had Valor SX, we had Valor plus Metribuzin, and we had Dual 2 Magnum, and then Broadax XC. So we recreated these chaff lines in the fall of 2020, and we seeded them with 36.7 grams of water hemp, which is a lot. And And then assuming a 35-foot combine head, we estimated that that would take 5.6 pounds of chaff per plot to simulate the effects. So these are data from Illinois. So this is our field site at SIU. And then Wisconsin is here. So we had more weed emergence in the chaff line than we did when there was no chaff on top of those seeds. So we created a good microsite, probably with plenty of moisture for those seeds to emerge through the chaff that we put down. So this is not entirely unexpected. I kind of thought that we might see more emergence, but it's interesting that that's not what everybody saw. So the University of Kentucky saw something completely different, and we're still trying to make sense of what happened here. But the University of Kentucky actually saw that their chaff lines suppressed Amaranthus emergence, especially in this dual 2 magnum treatment, but this looked a lot like their control. So even without herbicide, we could see that the chaff lines seemed to be suppressing emergence in Kentucky. We don't know exactly what's happening here, but it could be related to the carbon to nitrogen ratio of the soybeans that we used at each of our sites, right? So the Kentucky chaff did break down faster. So I've starred Louisiana here because Louisiana didn't see any significant differences in the effect of chaff or herbicides, but they had a really high degradation rate here. So you see these negative numbers that are representative of the rate of decay of chaff. If you compare Kentucky then to our other states, Illinois and Wisconsin, Kentucky had the next highest rate of chaff decay. So what we think was maybe happening is those seeds were tucked inside the chaff began to decay rapidly, and of course any decaying biomass around a seed is going to be really bad for that seed. So we think that there was an interaction here that probably suppressed emergence in the chaff line. So I saw Dr. Prashant Jha from Iowa State University present, his student presented these data at the North Central Weed Science Society conference, and when I saw his data, I knew that I had to reach out to him so I could share it with you. So he provided these slides to me just give you a heads up, he saw the same thing basically that Kentucky saw. So I thought that was really neat. You can see his chaff lining system here, the chaff coming out the back of the combine. That's representative of the density of water hemp that he was dealing with. Or I'm sorry, this is, I believe this is Palmer. Water hemp. So he had this level of water hemp infestation in his plots. You can see here this chaff line that is 18 to 22 inches wide by 6 to 8 inches deep. So he has water hemp seeds that are concentrated in the chaff line, and this is at the center of the combine here with a 35-foot header width. So this was really neat that he quantified this, and I think this is a good takeaway message. So he quantified the seed within this half-meter square area here. He found inside the chaff line there were 75,000 seed per meter square. Outside the chaff line, there were 200 seed per half meter squared, or per meter squared. So that's a huge difference. You're actually succeeding. If you can capture the seed in the combine and get them into the combine, then they come out the back and they're funneled into this line where at least you know where they are and you can deal with them. Whether you're suppressing them or whether you're encouraging them to emerge, your seeds are in one place within the field. So over time, if you can put your seeds in the same place, and chaff lining in corn is maybe a little bit different. I think we can figure this out in soybeans, but corn is going to take more research. But we put the seeds in the same place year after year, and we can begin to deal with them in different ways. So this could reduce the spread of resistance by more than 20-fold per acre, according to the data here from Iowa. So he then went into a rotational strategy, and he plants here his corn. You see the corn rows are on either side of the chaff line. You don't want to disturb the chaff line once you put the seeds down. And this is the cool part. This is what represents pretty much what we saw in Kentucky. So with the untreated, with no herbicide added, inside the chaff line and outside of the chaff line, you do see a little bit of a shift here. Inside of the chaff line, you see a delayed proportion of emergence over time. So the seeds are emerging from the chaff line more slowly, even with no herbicide added. But when you use a pre-emergence herbicide here, you shift that emergence timing even more to the right. So there's a positive interaction here between the chaff line and his herbicide treatments that continue to control and suppress water hemp. So he actually had a banded application and then a broadcast application. But you can see inside the chaff line here, number of plants per meter square. We're talking about in the untreated 123 to 213 inside the chaff line, compared to around 10 for both of those, with still inside the chaff line, but with a pre followed by post program. Outside the chaff line, he had many fewer seeds here, 40 to 52 now compared to 123 and 213. He only had, here you see, single numbers for the pre followed by post program now outside of the chaff line. So, in conclusion, I have no idea how I'm doing on time. 5 minutes, okay, great. So, we've been talking for a very long time now about how diversity is key. We've really been focused on the diversity of herbicides. You know, how we can use different site of actions, different active ingredients, and try to sort of slow the evolution of resistance that we're seeing in our cropping systems. Crop rotations are the basis of all of this, and we know that we have too few crops in rotation, but we don't have a lot of other options, really. I could tell you about the work that I'm doing with hemp. I actually took out these slides because I thought I was going to be over time, but we've figured out that hemp is highly allelopathic, and its phytochemistry actually, the compounds it produces suppress Amaranthus seeds. We know this. So we've done work to simulate what a chaff lining system in grain-producing hemp might look like. We've taken that hemp residue and we've simulated chaff lines, and we see much significantly reduced water hemp emergence in a hemp chaff line because of those allelochemicals. So there are ways to use allelopathy and those chemicals that cover crops make and other crops make to further suppress our problem weeds. But we have to figure out how to get more crops in rotation, and right now, being from Southern Illinois with poorly drained soils, I can tell you that hemp's really difficult right now, and we have a long way to go with our agronomy in Southern Illinois. So we know that we need to do herbicide mixtures with multiple effective sites of action. Effective sites of action meaning that we need to choose multiple sites of action with no resistances present in that field that we're applying to. But Aaron, if you were here for Dr. Hager's presentation, he talked about metabolic resistance. There's a lot of information about metabolic resistance, but we don't really know if this is still going to be true with the evolution of metabolic resistance. We think that right now a diversified strategy is the best, but we don't really know how the evolution of metabolic resistance has come about. So because we don't understand it, we hope this remains something that is true. We can see that IWM frameworks are really important. So we need to start adding in more diverse tactics. So we need to stop relying quite as much on chemical control. We need to keep using those tools, right? We don't want to ever put away a tool that's useful, that's working for us. But we want to figure out how to integrate other things along with it to sort of thin the herd so that we're applying those chemicals to fewer numbers of individuals. And that's where things like cover crops can come in. And then we need innovative solutions. I've shown you some things here that are, not realistic practical concepts for us today, but it takes out-of-the-box thinking. We have to get creative and try to figure out what other tactics we can use so that we can prolong the chemistries that we have. And it's all going to come down to knowledge of biology and ecology. So without knowledge of those weeds and what they do, when they germinate, how they grow, we're not going to be able to build effective strategies. So we have to understand the weeds that we're dealing with first. And that's going to allow us to build in new technologies. And there are all kinds of great new technologies that are coming down the pipeline, things like digital imagery analysis that are going to allow us to create prescription maps so that we go in and spot spray weeds in fields, and we can use less herbicides that way. But I would like to mention that we also can't rely on these new technologies. We can't rely on these techno fixes to come in and save us, because there are ways that those weeds are going to evolve to those technological management tactics as well. It's all about weed evolution, and if you don't understand the ecology and the biology of the weeds, you can't predict how they're going to evolve to the new technologies. So, and I wanted to say, so for a lot of really great information about herbicide resistance and how to manage herbicide-resistant weeds, Dr. Pat Tranel has a great presentation up about metabolic resistance if you want to learn more about that. And then thank you from me and from all of my lab for being here today and listening to our research.